Thank you for coming today. I'm really excited to be here. I was supposed to be one of your tours, but as Rafe mentioned, I work with mouse models, and the idea of taking all of you through a process of showering and getting into scrubs was not a realistic one. And so what I'd like to tell you about today is the power of mouse models in research. And so some of this might go over your head, but I really hope to bring home how powerful they are, how useful they are for complex models and disease systems like cancer, so you can appreciate what they can do potentially. So as you all know, you've been talking about a lot of genes today. Both humans and mice have lots of genes. And so when we started to look into this, we realized that we have tens of thousands of genes, well over 20,000 genes in mice and in humans. And genes contain these instructions, right? So I tend to think of genes as instruction manuals. And these genes tell the cells how to make proteins. Proteins do nearly all of the important tasks that we have in our cells to keep those cells working properly and to keep us as an organism healthy. And so this is just a depiction of that. There's a chromosome up here on the left. And if you were to unwind that chromosome, it's like a bead of DNA, a strand of DNA with beads of genes along them. So each of those genes, those 20,000 or so genes, each of them is an instruction manual for a very specific protein. And that protein has a very specific job. So how do we figure out what genes do? Initially, we didn't really know what genes were. We didn't know there were genes and there was DNA. It kind of came about as heritable traits, right? Like you would notice that a certain eye color might run in the family, or your uncle is especially tall and it runs in his family and his kids are tall. So there were things like that that we noticed in our populations and our families that eventually we were able to attribute to genes. But a lot of those 20,000 genes don't produce traits that we can see. They're a bit trickier to figure out what they do, and a lot of what we've been able to do is use animal models to try to discover or learn more about those genes and what they do in us. So some of the things that might come up in this, when you think about it, genes, our genetic makeup, might have an influence on whether or not we're lactose intolerant. It might affect our predisposition to have certain diseases or conditions, high blood pressure, heart disease, diabetes, as well as cancer. So as all of you are probably acutely aware, cancer is a disease of genetics. Cancers are often defined by dysregulation of genes, and that would mean when a gene starts to behave or produce that protein in ways that it shouldn't be doing it. The other way that cancers often present themselves are from mutations or variations of genes. So this would be consistent with these ROS1 fusions that we've been talking about. So when cancers are present, it's because of genetics, and that makes them uniquely or generally very amenable to study in mouse models. So there are lots of different animal models out there. These are some of the main reasons we like to work with mice. Their anatomy, physiology, and genetics are very similar to humans. They can be bred easily. They're small. They've got a nice disposition. And because of their short lifespans, it allows us to study diseases that progress slowly in humans in a very short time period, so we can advance the science more quickly. And one of the things that I think is critical to the value of mouse models is the creation of inbred strains, which I'm going to explain to you on the next couple of slides. So the history of the laboratory mouse: in the early 1900s, some hobbyists, including Abby Lathrop, which is this retired school teacher here, started to breed house mice together to get coat colors that they wanted. And so this was the first intentional breeding effort to alter the genetic makeup of mice. Somehow, these two researchers at Harvard, Ernest Castle and Clarence Little, learned of Abby Lathrop's little side job, breeding mice, and became interested. So they got some mice from her, and they took this idea of intentional breeding a step further. They decided that they would take small groups of mice, they would breed them together over generations and generations until they created what we call an inbred mouse strain. What's super exciting about inbred mice is that they've been bred together for so long that the genetics of these mice are identical. So those 20,000 genes in that mouse is exactly the same as the 20,000 genes in its brother, its sister, its mom, its dad, and its children. Excuse me. So in this way, we can use inbred mice as a palette, a baseline, to look at the role of genes. in the disease processes. And there's two main ways we do it. So you think you have your 20,000 genes in your inbred mouse, and now we can either add one gene or we can take one gene away. So this reduces a lot of the complexity, a lot of the background noise that you observe when you work with outbred populations like humans or wild animals. So I'm going to describe to you the way that we make gain-of-function mice, where we add one gene, and how we make loss-of-function mice, where we remove one gene, and how that can be helpful in studying diseases like cancer. So I'll explain the technology very briefly for each of these two approaches, and I'm going to give you an example of how these were used for some cancer studies. So the first transgenic mice that were made, these are gain-of-function mice, so you're adding a gene, were made in the early 1980s by the laboratories of these two gentlemen. And what they did was they decided to introduce what we call a transgene. A transgene has an on switch or promoter, and the gene that we want to have added to that mouse. And so what they did, and what we still do today, is a process of pronuclear microinjection. So what I have here is an image under the microscope of fertilized mouse eggs. And we have here, it's like a glass straw that we've polished and tapered and applied a vacuum to, that we can gently hold those mouse eggs. And then we're going to come in with a very, very fine needle and introduce a transgene, that DNA, that extra gene that we want to add, with an on switch. After we've injected all of our mouse eggs, our fertilized mouse eggs, we transfer them by surgery into foster moms. So surrogate moms is over here on the right, is a depiction of this. And she will have pups. And sometimes, if we're lucky, a certain number of those pups will incorporate that transgene into its genome, into its chromosomes. So all of the babies, the offspring it has, will also have that transgene. So in this way, we will now have a mouse that has 20,001 genes, for instance, that are being expressed, and we can compare them to the mouse that's exactly the same, but only has those 20,000 genes. So this allows us to put in sharp relief the value or importance of one gene in a complicated process. So this is the first transgenic mouse that was made. It was on the cover of Nature, I think, in 1982. This mouse on the left here has this transgene with the growth hormone. This guy on the right here is his sibling, same age, same gender, but he doesn't have the transgene. He didn't inherit it from his parents. You can see this puts in stark relief the power of transgenesis. This mouse is clearly much, much bigger than his brother. And the only difference between these two mice is that one transgene. So one of the early studies that was done in cancer was to address the role of oncogenes in mice. So as you all know, an oncogene is a mutated form of the gene that has this potential to cause cancer. And at the time, in the mid-1980s, the hypothesis they wanted to ask was, does oncogene expression in normal cells lead to tumor formation? So this is actually the proof that an oncogene can create cancerous tumor cells. So to do this, they've created several different mice, several different transgenic mice. The first one is our inbred mouse. He's got those 20,000 genes. Nothing has been changed in him. The second mouse they made had a transgene for an oncogene called MYC. The third mouse had those same 20,000 genes as his brother up here, but now he has an oncogene, that extra gene, for a RAS oncogene. And this fourth mouse that was made has those same 20,000 genes as the original mouse, the inbred mouse, plus both oncogenes. And then what they did was they evaluated groups of these different types of mice over a period of days, which is on the bottom there, going from left to right. We're seeing the progression or age of the mouse. And they look for the percentage of tumor-free mice. And so the inbred mouse, which is normal and healthy and immunocompetent, no tumors were detected over that period of 200 days. For the mice that had this extra gene, this transgene, for the MYC oncogene, they do start to show some evidence of tumors. By the end of the study, about maybe 80% of those mice remained tumor-free, 20% of them developed tumors. For this third group of mice that had a different oncogene, RAS, you see that the mice get tumors more quickly, and more of the mice have these tumors. And when you make a mouse that has 20,000 genes plus two transgenes, these two oncogenes, you see an even more dramatic onset of tumors in more of the mice. So what I showed you so far is the inbred mice. These 20,000 genes are fixed, and then we can add in a transgene to see what that extra gene can do. And now I'm going to tell you about how we can remove a gene to see how that mouse behaves when he has that 19,999 genes. And this will help us evaluate the role of one gene in a complex system or disease. So knockout mice used to be really tricky to make, as Rafe can attest to because we've done this for him. It takes a lot of time, but in about 10 years ago, CRISPR-Cas9 came along, and this is something that you may have heard about. It's a technology that enables us, excuse me, to edit genes very easily. So these were the 4 main scientists that were involved in developing this technology, or providing an application for this technology, and it resulted in 2 of these individuals getting the Nobel Prize in Chemistry in 2020. CRISPR-Cas9, in this most simplest form, is extremely powerful because these are engineered DNA scissors. So I can go into that long chromosome, I know where my gene is, I know the DNA sequence of that gene, and I can design scissors, these CRISPR-Cas9 scissors, to cut the gene out. So what I'm going to do now is I will take my fertilized mouse egg again under the microscope, and this time I'm going to put in those DNA scissors just to cut out that one gene so I can see what it does, or what it doesn't do, what happens when it's not there. So this is an example, done the old-fashioned way, of a p53 knockout mice that was made. Roughly half of human cancers have mutations in p53, and so the first question that scientists wanted to ask in a model, a mouse model, was what happens if we remove that p53 gene? If we take that out, how does that knockout mouse, that mouse that doesn't have p53, behave compared to its counterpart, the inbred mouse that has 20,000 identical genes instead of the 19,999 genes? So this is a very similar kind of plot. We have on the axis on the bottom, we have days or time. And what we're going to do is we're going to follow these inbred mice and compare them to the p53 knockout mice that are only missing that one gene. And we're going to track them for the percent of the animals that are tumor-free. And you can see that those inbred mice remain tumor-free for the duration of the experiment. And the mice where only that one p53 gene has been removed, they start to develop tumors, and they develop them very quickly. And at the end of their study, at the end of this period of time, about 40 weeks out, all of the p53 knockout mice have developed tumors. And this is a very strong way to demonstrate that p53 is a tumor suppressor gene. So to summarize, I've given you some really global ideas about how we look at transgenic and knockout mice. The mice that we're making right now for Rafe and Lynn are basically transgenic mice, where we introduce those ROS1 fusions, and then they can compare those mice to those inbred mice that have 20,000 genes but not the ROS1 fusions. And by doing this, they can either look at them from a very basic cancer research point of view, which is usually where we're at, where we can look at and study different aspects of cancer from the early studies of testing hypotheses. Is this gene fusion imperative? Does it drive cancer tumor development? Or later in development, can we use these mice and test potential therapeutics? Can we mitigate or improve outcomes by certain types of treatments in these mouse models? At the same time, in translational work, we can actually use these genetically modified mice to go through a more active process of trying to get an application for the science that is generated from the basic cancer research. So that's all I have for you today. Thank you.